everyone, I'm Angelina and I'm a nursing major, and today we will be discussing memory formation and memory transfer. So, the reason why I chose to research this topic is because while watching a lecture video regarding axons of squid, they mentioned that researchers use sea slugs in order to study memory formation, and I was like, sea slugs are super cute. So that's why I chose to research this project.
Hi, my name is Pia Dineri, a nursing major. So, if there's any viewers in here right now thinking about focusing on memory enhancement studies, hopefully this podcast could spark more interest and wisdom to pursue that dream. And maybe he or she can one day open doors to new ways of overcoming traumatic memories and restore the ones worth keeping.
My name's Viviana. I'm a kinesiology major, and kind of one of the reasons I found interest in researching this podcast is because I have a terrible memory and I forget everything. So who knows, maybe I could freeze my memory and transfer it to do good for me or something. I don't know.
Honestly, a great idea.
Wait,
It's a really good idea. I'll do that too. I'll transfer someone's memory to mine and now I don't have to study. It's all good.
Exactly.
Great idea.
My name is Lisa Chang. I am also a nursing major, and I don't find sea slugs cute at all. I think they're pretty weird. I'm sorry, Angelina. I know you think they're cute, but I find them pretty weird.
No, you're good.
They're really slimy, so I don't know if I think that qualifies as cute to me. But why I am interested in this topic is because it would possibly help me better understand memory myself, because I find myself going in to different rooms and leaving them because I have no idea what I'm looking for, or I forget within that moment what I went to go grab.
Alright, so as we all know, memory plays an extremely integral role in our abilities to survive. Not only do we use it to memorize phone numbers, birthdays, and due date assignments, but memory is also pertinent to the process of ingraining some of our most fundamental abilities, such as writing, talking, and our reactions to certain stimuli. So what we aim to discuss today is how memory is formed in our brains. And just to get the fundamental idea of memory formation within the cellular level, we will be taking a look at sea slugs and how they develop a learned response to certain stimuli.
For us to fully understand sea slugs, we need to learn a little bit of a background about them. According to a professor, psychiatrist, and a Nobel Prize winner, Eric Kandel, sea slugs plays an essential role to neuroscience and to the expansion of it. In today's research, we will be discussing a particular kind of sea slug called Aplasia californica. Aplasia is the term used by scientists to identify medium to large-sized sea slugs. One that is particularly essential to neuroscience is found in the coasts and gulfs of California, hence the name Aplasia californica. Aplasia californica comes in 3 different colors. They can be found red, green, and brown. One picture I can show you is this one here. This one in here, this Aplasia californica is red. Depending on
you know what that reminds me of? It looks like boba.
You know,
it looks a lot like boba.
Oh wow. It does. Wow, I didn't think of that. It's because of the color of the algae that they eat. So if they eat a green algae, their color is going to be a little bit more on the green side. If they eat the red ones, the color will be more like this, which is reddish brown, I guess. So it depends on the color.
Is that the normal size?
No, actually, no, this is not. They can be bigger. They could go up to like,
wow.
This is probably medium size for them, but they can easily grow up to 16 inches from here to here. 16 inches
wow.
there and 8 inches up here. This is probably considered small or medium, and they can weigh up to 35 pounds. right? So I would say pretty big for something that's just swimming around under the sea. Now we have to learn a little bit about their anatomy and physiology. Right over here, if we look at this image, we can see that there are a little bit of tail in the back, foot, and parapodia. These three structures are responsible for helping them in locomotion and swimming so they can move around under the sea. And they also have the gill for them to breathe. For them to get rid of their waste, they have the siphon. For them to adapt in their environment, they rely on mantle up there. This is the mantle. Well, that mantle is like a cover or a transparent shell. It helps them secrete a purple dye, just like this one in here. The first picture that we had right here. This particular Aplasia californica is producing that purple dye. Whenever they are handled or disturbed, this is one way for them to scare away their predators so they can escape. So sometimes it can be toxic, sometimes it that. It depends on the concentration that they have. But luckily, according to International Union for Conservation, or IUCN, their status is safe for now. They're not extinct or they're not on the red list, meaning we can continue studying them and improve scientific research without hurting their species or being destructive to them. Well, as long as we follow protocols, environment policies, and wildlife conservation standards.
Yeah, so why do we use Aplasia californica in like research regarding memory formation?
Yeah, actually, I have another one to show you that I have to mention. Very, very important. Well, Aplasia californica, they have been laboratory specimens for so long. And up to this date, scientists, they still rely on them. On this part of their body, this is where most of the synapse activities are happening. They have neurons that are so large and nerve cells that are so big that these scientists, they can easily figure out which of those neurons or nerve cells are responsible for what particular behavior, right? Or this neuron is probably particularly responsible for learning. This could be the one that is responsible for memory. If so, is it the short-term memory or is it the long-term memory? So they're very blessed with those neurons, though they're not a lot. And these neurons, they continue to grow throughout their lifetime and making them really a good ideal candidate for studies like this.
Yeah, so as Pia had stated, memory can be traced by looking at the activity of certain neurological pathways. And before we discuss the actual mechanisms of memory formation, I think it's important to first understand that memory is just not concentrated in a single area of our brain. It's more sort of the integration of these networks of billions of neurons. So memory relies on the strengthening of the synapses along certain neurological pathways within that network. And the easiest way for me to explain this is to give you guys an example with two neurons. Axon terminals connecting to the dendrites of the next neuron, and so on. All right, so when we're attempting to store information like a phone number, what we usually end up doing is repeating it. So we will often repeat the phone number several times. And this is an attempt to ingrain it into our brains. And through that repetition, the neurological pathway will experience a very frequent stimulus running from sidon through the axon all the way to the axon terminals and dendrites of the next neuron. And what researchers in collaboration from the European Journal of Neuroscience found is that the frequency of the stimulus or the action potential can strengthen the
synaptic connection by increasing the neurotransmitters as well as the receptors along the dendrites. So the stronger the connection between the synapse of the neurological pathway, the easier it is for us to recall a particular piece of information. However, it is also possible for us to lose this connection. In fact, it takes only 30 seconds for a synapse to revert back to its original state, which occurs when there's no longer a frequent stimulus running through the neurological pathway. And we see this mostly in relation with short-term memory, where our brain only temporarily holds on to that information. And within a minute or so, we can't seem to recall it. So this idea that active neurons will create stronger synapses and inactive neurons can lose connections is called neuroplasticity. And it was discovered by Jerzy Konarski in 1948. And this becomes the basis of long-term potentiation or long-term memory. And we see this particularly with studies of musicians, and especially the brains of violinists are extremely interesting to look at when talking about the neurological pathways produced. So Bruce Wexter, a professor and senior scientist at Yale, found that the right motor cortex of most violinists was significantly larger than their left motor cortex. And this is due to the fact that violinists will often use their left hand to play the strings. And when they're playing that, it's all about memorizing these precise movements of their fingers. And so this translates physically by the significant growth of their right motor cortex. And this brings up the question, where is memory stored? Well, researchers from our slug experiment believe that RNA can be the key to memory storage. It's more based on the idea that DNA and RNA codes for the production of more neurotransmitters and more receptors. So the ability to strengthen a neurological pathway is coded in DNA and RNA.
So like Angelina has stated, RNA does play an important role in the process of learning and memory formation. And so we are going to be looking at the experiments that had helped us better understand those RNA and memory formation. So the first experiment is going to be carried out through a researcher named Glasman, who had used sea slugs to see if they could learn through shock trials. So how he had done this experiment was that he used one group to receive shocks and another group to receive taps. And after some time of having the sea slugs understand what those shocks and taps had meant, they had then extracted RNA from the nervous systems of both the shocked and the tap groups and injected it into a controlled group of sea slugs that hadn't done any shock or tap trials. And they had injected the RNA into 7 of the controlled sea slugs, and the other 7 did not receive anything. And so after injecting those sea slugs, the team had then tapped their tails to see how they would respond. And the RNA injected sea slugs had basically responded exactly like the original group had responded. So they had then recoiled for about 40 seconds as though they were transferring memory from one sea slug to another, which I guess you could say it was their learning process through the synapses and the neurological aspect of understanding what those shocks and taps had meant.
Yeah, so this experiment was really interesting because it showed that not only are learned behaviors or memories stored within RNA molecules, but memory can also be transferred from one specimen to another.
Yeah, exactly. So after that, another research was conducted through scientists of the University of Chicago. They had used mice in the same exact type of way and experiments, but they were focusing more on a protein, which would be a protein from the YTH family, more specifically known as the YTHDF1. And the YTHDF1 is used to recognize a modification on a messenger RNA in mammals that will be called N6 methylidosine, or known as M6A for short. And the M6A plays an important role in the process of learning and memory formation. And so having this protein, the YDHDF1, recognize the M6A modified mRNA, then promotes their translation to proteins, which then demonstrates how the translation increases specifically in response to the nervous system stimulation. And so having researchers look at the mice, they had conducted the same experiment in a way. So they had a group of mice with the protein of the YTHDF1 and a group of mice without it. And so after conducting the experiment with the mice that did carry that protein, they then extracted the RNA and the protein and put it into the mice without it. And so putting the YTHDF1 in the mice that didn't have it, they were able to see that the performance on memory and learning tasks had improved dramatically, the same with the sea slugs. And so you could see that the protein can respond to neural stimulus, which could contribute to controlled translation. And even though neuroscientists would agree that memory particularly lives in the synapse, especially long-term memory, after cross-examining the study of Glassman's and the study at the University of Chicago, it would suggest that the nucleus where RNA carries out its DNA in transcribing would possibly be the key to decoding the memories that are stored. And this can then help us understand us as mammals and humans in real life situations on how memory works.
All right, everyone. Now that you guys know a little bit more about sea slugs and the idea of memory transfer from my lovely podcast teammates, I want to talk about how all of this stuff can possibly be applied to real life situations. Some of our listeners out there might have an individual that they know, friend, family member, friend of a friend that they know that struggles with Alzheimer's, Parkinson's disease, a brain injury, an addiction, something along those lines right there. And they've seen that the negative effect that it has on that person. I mean, the idea is to start off with memory transfer, but the thing is, it could lead to so much more. To begin with memory transfer, how cool would that be if we get that down and eventually we could transfer our own memories to our grandchildren, great-grandchildren. They could kind of see what we see, you know? Any conspiracy theorists out there has the idea that Walt Disney's head's frozen under Disneyland. Big Disneyland fan myself, so I mean, if it is frozen under there and somehow his neurons are fully intact and they're all good, maybe we could see some of the memories he has. I mean, it's a long shot, but who knows? But the real thing I want to talk about is Alzheimer's and how this idea of memory transfer could possibly lead to a cure to some terrible diseases. Alzheimer's is that one I kind of want to talk about in particular. In a study posted by Ohio State, they have an article out there called New Thoughts About Brain Disorders. And in that study, it's very similar to what Lisa was discussing with her mouses in that one, where in this one, for instance, they actually take sea slugs and they extract neurons out of them. They're very similar to how human brains are set up. I mean, sea slugs only have 20 million neurons, where humans have, I think it's about 100 billion. So, I mean, definitely quite a big difference between the two, but brain similarity setup-wise that way. What they're hoping is, since sea slugs have so little neurons, they're hoping that by being able to figure out which one is like kind of that bad seed in there, by being able to extract it and fix it, they're going to kind of, their words, they say hypnotize, but I guess I'll say pretty much tune it up, make it good as new, and then reinserted in that brain, they're hoping that that's that cure right there. And in terms for humans, I guess you'd say we're hoping that we could do that with memories and curing disease. Just recently, the Alzheimer's Association were saying that 1 in 3 seniors were dying with Alzheimer's. So this is just a start. Maybe memory transfer could lead to so much more. Maybe it could cure diseases. Maybe it'll give us a glimpse at what Walt Disney had when he first opened the parks. Or who knows, it could honestly lead to so many other things.
Yeah, so the advancement in our understanding of memory formation and memory transfer has so much potential to treat patients with neurological issues and neurological diseases. And one of the possibilities for a future method of therapy or method of treatment would be dealing with people with Alzheimer's, as Bibiana has mentioned. And what we could do possibly is have them receive treatment to help maintain and strengthen their neurological network of neurons. And hopefully that will allow them to retain more of their memories over time. Another possibility would be to help people struggling with addiction, and they would be able to receive treatment therapies to reroute their neurological pathways and help them overcome bad habits. Overall, I believe that these studies are just breaking the surface of what can become a new method of therapy or a new method of treatment. And I look forward to all the new studies and new advancements that will be made within this field.